Every cell in your body carries a story, a precise sequence of divisions and transformations that began with a single fertilized egg. For most of human history, this story remained hidden, an invisible choreography too complex to observe. John Edward Sulston changed that forever. Born in 1942 in Cambridge, England, Sulston grew up in a world still reeling from war, yet brimming with scientific possibility. The structure of DNA had just been discovered, and biology was entering a golden age. Sulston's path would lead him to become one of the architects of modern developmental biology, a scientist who would literally watch life unfold, cell by cell, under his microscope. Sulston's breakthrough came through an unlikely subject, a tiny transparent worm called Caenorhabditis elegans, barely a millimeter long. This humble creature became his window into understanding how complex organisms develop from a single cell. The worm's transparency was key. Unlike most organisms, every cell inside C. elegans could be observed in real time, making it the perfect subject for tracking cellular development. In the 1970s, Sulston embarked on a monumental task that would consume years of his life. He decided to map every single cell division in the developing worm, from fertilization to adulthood. Hour after hour, day after day, he sat at his microscope, watching cells divide, migrate, and differentiate. He tracked each cell's fate, noting when it divided, where it moved, and what it became. The result was extraordinary. Sulston created the first complete cell lineage map of any organism. He discovered that the development of C. elegans was remarkable 59 cells in the adult hermaphrodite. This wasn't random chaos, but a highly orchestrated program written into the organism's genes. One of Sulston's most fascinating discoveries was programmed cell death, also called apoptosis. He observed that during development, exactly 131 cells were destined to die at specific times and places. These deaths weren't accidents or failures. They were essential parts of the developmental program, sculpting the organism like a master craftsman, removing excess material to reveal the final form. This revelation transformed how scientists understood development. Cell death wasn't just something that happened when cells were damaged or diseased. It was a fundamental process, as important as cell division itself, programmed into the very blueprint of life. The genes controlling this process in the worm turned out to have counterparts in humans, opening new avenues for understanding cancer, neurodegenerative diseases, and aging. Sulston's meticulous work laid the foundation for the next great challenge, sequencing the worm's entire genome. The achievement was a crucial stepping stone towards sequencing the human genome. Throughout the Human Genome Project, Sulston emerged as a passionate advocate for making genetic information freely available to all researchers. He believed that the human genome belonged to humanity, not to private companies seeking to patent genes for profit. His principled stance helped ensure that the human genome sequence would be released into the public domain, accessible to scientists worldwide. In 2002, Sulston received the Nobel Prize in Physiology or Medicine, sharing the honor with Sidney Brenner and H. Robert Horvitz for their discoveries concerning genetic regulation of organ development and programmed cell death. The recognition celebrated not just a single discovery, but decades of patient, painstaking observation that revealed fundamental truths about how life builds itself. Beyond his scientific achievements, Sulston was known for his humility and his commitment to open science. He worked in a collaborative spirit, believing that scientific knowledge should be shared freely rather than hoarded or commercialized. His autobiography, The Common Thread, detailed not just his scientific journey but his ethical battles to keep genomic information in the public domain. Sulston's legacy extends far beyond the worm he studied so devotedly. The techniques he developed for tracking cell lineages are now used across biology. The concept of programmed cell death he helped elucidate has become central to understanding development, immunity, and disease. The genome sequences he fought to make public have accelerated medical research in countless ways. John Sulston passed away in 2018, but the map he created endures. Every time a researcher traces a cell's lineage, studies apoptosis, or accesses freely available genomic data, they walk a path he helped clear. He showed us that even the smallest organisms can reveal the deepest truths, and that the patient observation of life's details can unlock its grandest mysteries. In decoding the development of a tiny worm, Sulston decoded principles that govern all complex life, including our own.